All right, let's get started, guys. The quicker we do this, the quicker we get to the weekend. So first, I've been talking about getting the human genome, but actually we're talking about sequencing technologies. So I realized this is something that I didn't kind of emphasize. Why am I using this title? I'm using this title because the technologies that are shown in the past lecture and today's lecture were used and are used for sequencing the human genome. You'll see that the filling the gap was done by a technology called PacBio, Illumina, sorry. It was done by Illumina, and so these technologies are still relevant for getting the human genome, and obviously it's being sequenced more and more. And so if you remember, we talked about the Sanger, the automated capillary Sanger dideoxy sequencing methods by Applied Biosystems. So that was the first technology that was used. Now there's different ones which we'll be taking a look at. Okay. Before I get into that, just a few updates. Remember that next week on Friday, you'll have to submit homework 4 and the essay. And then you have reading week followed by the exam on the 23rd, which is a Wednesday. The exam is up to this lecture, so today's lecture. So this way, because I know you have an assignment to submit, an essay, you have to figure out the paper, and also you have an exam coming up. So what I'm going to do is just stop the lectures for the exam today, and whatever we cover next week, we'll do it after. People ask me about questions that could be on the exam. So the exam is 40 points. This is one of the questions that I actually had in the exam, so I'm going to have to make a new one. But it's along the lines of, it's 1990. You have been put in charge of determining the genome of a double-stranded RNA yeast that is approximately 12 million base pairs, that has 10 chromosomes. So how can you do that? So provide a description of the approach you'll take to kind of sequence it, okay? So the exam is in the form of short answers. So it doesn't mean that you'll have 4 questions that are 10 points each. Some of them are smaller, some of them are longer. This is one pretty long one. I think it's the longest one that was there in the exam. When you see 10 points, don't give me just two steps, okay? Get into the detail. Every detail you're giving me that is kind of a major step is probably going to be a point. So in this case, what you would answer, you would go through all the steps. Karyotype, you would explain how you would karyotype, what are the results from that, and then you would characterize the genome. You would break it down into smaller pieces, build your library based on this amount here and this amount of chromosomes, etc. Does that make sense? Just like we did in the lectures. I might ask you stuff about vectors. What type of vector would you use for this, and how would you do a functional assay with that vector, things like this. So it's based on what we learn, but it's kind of combining multiple things at the same time. It's not like what is a shuttle vector, right? So it's more like how do you use everything that you learned together to give me an answer for this. Does that seem fair? Easy? So this is the past 5 lectures, I guess. You can just summarize them in 2 sentences or 1 sentence each, okay? One important thing for this, and I've seen this in your assignments, so I have to cover this up. In the exam, I'm giving you a specific space to talk. One mistake that was done a lot in the writing is not being concise. Not being concise usually comes from either the English not being the first language, or just saying many things repetitively without giving actual substance, okay, being too vague. So you have to be concise in the sense that we're not having a conversation really. It's more like you have the genome that is double-stranded RNA. Okay, first step is separate it so that you have 1 strand, right? Or cut it into pieces. So I have not get into the full introduction of, try to be concise. Adding more words does not really add to the answer. And because it is a writing, why am I saying this? Because it is a writing exam, you don't want to end up not having enough time. Because choosing A, B, C, and D, that's easy. You just read it, whichever one makes sense to you, you circle it. But in this case, you have to formulate your ideas in your head and then put them on paper. And sometimes you will start writing and figure out that you have a mistake or something in your logic and then have to erase, etc. So it's important to be concise so you don't waste time. Okay? This is relevant for the midterm and the final. For the assignments, the problem with not being concise, if I actually apply the limit of words like that I have assigned, people did not respect that. They went up to 1,000, 1,500 words in the assignment. If I apply it, then you end up not having enough information because you've been talking about the same thing for 3 paragraphs, right? So it's removing the work from you, but it requires you to think a little bit more to make these concise sentences and speeds up your writing, okay? So this is easier for you, easier for me correcting, and does not make the reader, does not get the reader lost, okay? Usually you put 1 information per sentence, that's it. Don't take 1 information and spread it on 3 sentences. Like, there's no point, okay? So this is one of the tips that I can give you for the exam. Obviously study the lectures, understand the concepts, but not understand them so that in the exam you will tell me this is a shuttle vector. More like how do you put them all together? Just like I formulated the class where I'm giving kind of a historical perspective, and so it gives you kind of a story. So in here I'm expecting a story kind of. I will do this, followed by that, get that, until I reach the sequence, okay? If you see 10 points, try to get about 10 informations or something like this, okay? So that's that. Doesn't really matter which informations you get. Obviously, there's 4 major ones that are in, you know, that picture where I have step 1, 2, 3, 4. Those are necessary to be there. But then obviously, I'm not gonna penalize you because you don't have the one that I'm expecting. You can, you know, depending on whatever you structure your text, I will give you points for it. So it's somewhat open-ended. So this is something that's good. Is that clear? Okay. For the assignment, this is assignment number 3. So I divided it into 5 categories where I graded the assignment, the clarity of your position. I went to the supporting evidence and scientific argument, refutation of the opposing view. As I told you, tell me the other side and say why you don't like it. Critical thinking, this is just the general thinking through your text, and then obviously the communication. This point here counts for 2 out of 10. So one thing that I realized is that students, I'm not sure if it's the question that was formulated this way that made you maybe distracted you from how to answer it. Usually you get all your points, you lose 0.5 because your position is not clear from the beginning. So if you're trying to argue something, it's important to get your position right away and then support it with the arguments. Not tell me the arguments of each side and then tell me I agree with this one because it's more scientific. Saying that it's more scientific doesn't tell me if you agree with the science or not. It's just telling me that these guys are saying this and I feel like this is better than that. So that's not what is expected. It's more like I don't agree with this theory because research has shown that this is not true, this is not true, and this is not true, and these make sense due to history, etc. Okay? So start your text usually with your position, and you have to make it clear. Most students had it clear, but you had it clear at the end. So you would go through the whole overview of the field, let's say, of the 2 sides, and then you would conclude by, I agree with this view because there's a scientific consensus on it, although we will never know because it's still being researched. That's not telling me that you are critically thinking about it. That's just telling me you took people's ideas and you agreed with them, but you have to tell me why you agree with them, okay? By the way, this assignment, the corrections, I did not go like crazy on the grading. If you apply these, I don't even think that people will get below like 85, okay, in the class, like very clear, okay? So that's the first thing. Second, for the supporting evidence and scientific argument. This was out of 4, so actually I was impressed here. I've seen some stuff that I didn't even know myself, and I had to research to make sure. But you were very good at providing supporting evidence, especially for the natural origin theory. I've seen some other theories in there that were interesting, that 1 or 2 students had. So you were pretty good at doing this. Just keep doing exactly that. One thing, though, is that when you make up claims, let's say, for example, you're saying that the bioweapon theory is not true because some genetic features have been observed or the lack of genetic features observed does not represent genetic engineering or whatever. Tell me what the genetic features are. You know, this is, be specific, because these abstract claims, you can make a full text about this. I can literally make a text that is fully abstract, vague, that talks about genomics, genetic features, and because I have an idea about it, right? I have an idea about the field, but I need specificity. Why specificity? Because it tells me that you actually understand what is happening under there and not just talking about genetic features. Everything has genetic features. And some look like this and some look like that. And when you're specific, then you know what you're actually talking about. For example, one genetic feature that you could talk about is these recombination sites, or these sites where enzymes would cleave. So when the enzymes cleave, they leave these traces, these sticky ends, right? And so these are genetic features that were not found in the virus. Then you can talk about that. But saying genetic features have been observed, what is it? Like the nucleotide set, which is ATGCG, what is it exactly, right? So that's that. Depth. So depth, this is one thing what I understand, because scientifically more has been, it's easier to prove the theory or to support the theory that it has a natural origin. But that does not mean that there's no research that has been done for the opposite side. So you had a hard time counter-arguing the bioweapon theory. And usually when you got to the bioweapon theory or the lab leak theory, it turned more into philosophical and political than scientific. But you can test the bioweapon theory. For example, a bioweapon, what does it need to have as a feature? It needs to be highly infectious. It needs to cause damage. It could be specific to a certain subset of the population, for example, the elderly. And so if you were to engineer a bioweapon like this, then you would try to augment those features. And there's research that has been done to show that, no, that was not done. Obviously, you can tell me that the world has been affected by this virus, and so because the whole world is affected, then how can it be a bioweapon? Or saying, for example, China created the virus, but they were the first infected, so it doesn't make sense that it is a bioweapon, because that's basically attacking yourself first. Of course, but we're in a genomics class, so tell me about the science. So people talked about the spike protein, the furin cleavage site, which were thought to be actually modified, and those are potential modification sites that could make it into a bioweapon. Some people talked about past bioweapons that were created and made kind of a parallel to it, like compared them both. So these were interesting, but just try to remember that, yes, the political side, philosophical side are interesting. The feeling side is interesting too, but don't forget the science, okay? So that's that.
It says graded, but the grade hasn't appeared yet.
That's what I tried to do this morning, but I think don't know how to use Moodle properly, so I have to figure out where the hell is the grade, okay? So I'll take a look at that after class, okay? Refutation of the opposing view. This is interesting. Some people wrote their article directly just refuting the bioweapon and forgot about the other side. It makes sense, it works. So you can debunk the bioweapon, which is something that's interesting, but tell me what about the other side? I need to see the two sides, okay? Because telling me that one side is wrong does not automatically make the other right. And if you like if I tell you, I don't know, I like apples, doesn't mean I don't like bananas, right? Makes sense. So tell me why the hell this is wrong and why the other one makes sense. So some people just went through the against the bioweapon or lab leak theory and then forgot about the other side. And because they were refuting these two, their refutations, as I just said, were not based on evidence, and were based mostly on kind of logical thinking, right? Why would you have a bioweapon and be the first one to be targeted by a bioweapon? Or the lab leak theory, which was then debunked, and they found that actually it was a mistake, there was a lab leak. So if you search this, you'll see that there was a lab leak. What else? Again, the side that had data on it, a lot of data on it, you were pretty good at doing in here. The opposite side, it became a little bit more vague. So when you're refuting the the opposing view, you had a harder time to be more specific than if you were talking about the zoonosis, spillover, or whatever, okay? I agree with this theory and not that because of overwhelming scientific consensus. Okay, that makes sense. You follow the experts. But what's telling me that actually just because there isn't research on the other side, that the other side is not true, right? It's not because you see scientific consensus on something that means that it's the only potential reason, okay? So this is more just of a thinking. Also, you're a scientist too, so forget about scientific consensus, think about yours too, okay? You have the capacity to do that thinking. So this usually occurred when you had presented both sides and the claims they were done by both sides in the scientific or non-scientific community, and then at the end told me your stance. So when you told me your stance, the only thing that was left was, I agree with this side because there's more consensus on it, but no. Tell me what you think, why you think it, and why this is wrong, okay? Not because of the scientific consensus. Because you believe that 96% of the coronavirus that happened in 2020 is similar to the RAG-13, the RAG-13 variant of this virus, and so there's 4% that is left, and that 4% could be potentially engineered. But if you take a look at the mutations where they happen in that region, it's more random than specific. Things like this, right? And you have papers, actually, you could use just one paper to talk about this, because there are scientific papers that were published, and you can find them on PubMed, that all they do is exactly this assignment. So they go through all the theories, and then kill them one by one, and then explain which one that makes the most sense, okay? So with a little bit research, you can talk about the bioweapon, lab leak theory, whatever theory there is. Critical thinking, actually this was not bad. You guys were pretty good at this. Most people got full points for this. You were able to talk about both sides, except for the people that took one direction and then debunked it and forgot about the other side. But in general, you were able to present both sides, and you had some sort of scientific reasoning. You were able to assess both hypotheses, most people, and then tell me which one was right. Some people even went further. For example, some students showed even more higher critical thinking. I can't give you more than the points that you're given, but talked about some philosophical concept like the Occam's razor, which says basically the least amount of assumptions is usually the right explanation. So you could use philosophical things like this, right? This is actually pretty interesting. I had another student who asked me a question at the end, more political and less scientific, but I replied to that question. So these are things that you can do to make your text a little bit more interesting, asking questions to continue, for example, the conversation. Now, obviously, this is like additional stuff, okay? I'm not basing it on this. This is what I base the critical thinking on. But, free for all. You can do whatever the hell you want here. Clarity of communication. Let's see. Yeah, so the main thing that I so everyone has pretty good English, everyone has pretty good writing style. You had a good structure. You present one side, the other side. The intro is to give background on what's happening, or you give your position and then get directly into the argument and then say this is wrong because this was shown like this, etc. Some students had vague phrasing, so please try to avoid this, because it just shows me that either you don't really have an argument, and I don't want to offend anyone, I'm just trying to think it through so that we can improve here. Either you have no idea about that and you're just trying to find some sort of logical argument there. If you see yourself in that position, avoid that. The way you solve it is find a reference for exactly what you're saying and go use that. That will remove the vague phrasing. You can paraphrase it, and when you paraphrase it, it starts sounding like if it's your own thinking. Okay? There's these ways of playing with the words. Grammatical issues, very minor, nothing too crazy. Long sentences, you try to be concise, but this is hard to do, right? So long sentences, because when you're having an argument, scientific argument, sometimes you try to include 3 different things in the same thing to bring your idea. So this is not as bad, just the vague phrasing, please try to avoid that, okay? What you can do, though, is you can break them into multiple sentences and try to transit better between the ideas, right? So, first, second, third, and this is what you were talking about in the beginning, okay? Obviously, this was not all in the assignment. Didn't give you a full do this, do that, but that was the main exercise of this. It's to see how you guys are actually doing from the get-go, and then to actually start improving for the rest of the semester. So this is what I realized. I think the average is like 85 or 80 or something like this. If you implement these in the remainder of the semester, you should be good. Should be very good, actually. Okay? Easy. If you want to argue about your grades, we can do that. I graded them twice, so I'm pretty sure what I'm doing. But you could try. You could try. Okay. Last class, Niti made me realize something here, that I misexplained this, and I don't want you to get confused. So I'm going to get back to it. We were talking about the pyrogram for the pyro sequencing, and I was telling you that when the nucleotide gets added, depending on the amount of nucleotide, at some point you reach a maximum signal. And then the student asked me, well, if you're adding nucleotides, then technically, if you have multiple Gs here, they should all have a single peak like this. So how are you saturating the machine? Does that make sense as a question? Okay. Remember, pyrogram, you're adding one nucleotide at a time. So if, for example, you add one nucleotide in your picotiter plate, and then that nucleotide gets attached 5 times in a row, it will only appear in this place here. So you will have this peak, double if there is 2, triple if there's 3, etc. So that does not mean that the G here, so in this case, you have G, C, T is not clear. There was no signal in the flow, right? So you don't see any T. Then there was 1A. Then you get to G. This does not mean that there was only 1 G. There was 2 G added here because you have the first one here and the second one here. Okay? And then you have 2 Cs and 2 Ts. So this is the nucleotide added to the picotiter plate and not to the sequence. To the sequence is the height of the peak at that specific time. Cool? Okay, almost done with this topic, which is actually pretty cool. The new technologies that are coming out. So we talked last week about pyro sequencing. We talked about also solid sequencing by applied biosystems. Now there's also other technologies that exist in the realm of second generation sequencing. The top commercial platform is the Illumina technology. This is the one I'm using right now in my own research because it's just super simple, okay? And it has a lot of advantages. It's quite precise and can be super fast in sequencing. So you have a video here that you can watch to see how this is done. But basically, Illumina sequencing starts exactly the same way as the other methods. You first have to fragment your DNA into these specific sized fragments. Usually there are, I think, 300 to 600 base pairs. And you will also add these linkers at the end. You always have to remember that when you're sequencing DNA de novo, you don't know the sequence. And the way to go around this is to add linkers to both sides of known sequence. And this you can do because it does not depend on the sequence, but it's just basically polymerizing these adapters at the end, at the 3' end. Okay? So when you fragment the DNA, it does not come perfectly as blunt ends so that you can add these adapters. Instead, it comes at these not sticky ends, but these weird broken ends, and so you have to repair them so that they are flush. They are exactly blunt, okay? And you do this just by adding the other, the missing, what do you call them? The missing nucleotides. You don't really need to know the sequence for this. All you have to do is just add, ligate those nucleotides, okay? And then after that, you will be doing an adenylation step where you're adding a single A at the 3' ends. And this single A will be useful to add these adapters, okay? Because these adapters will recognize that A and bind to it. So you can imagine these adapters as missing one nucleotide, which is basically the T that will bind to the A. So this is what I said here. And now there's different types of linkers that can be used for sequencing. We'll start with a simple example, and then I'll give you other examples that are used, which are for different forms of sequencing, okay? So the adapters, there's two of them that are used, still the same ones that are used. They're called the P5 adapter and the P7 adapter. And if you're doing a single read, so this single read means that you're reading only in one direction, then you'll have your sequencing primer attached to the P5 adapter. So that means there's nothing here. And then what you will do is that you'll add a primer that is, so this looks like, initially you don't have this orange line here. What you have is the double strand here and this small part here that is lighter, let's say yellow. And then when you want to sequence, you'll be adding a primer for that yellow sequence, which you know. That will bind here, and then you can use polymerase to come here and start sequencing, okay? If you remember correctly, second generation sequencing depends on PCR. So it requires amplification of the sequence. This is why you need these primers. When we'll take a look later in the lecture at third generation sequencing, you'll see that we'll get rid of all of this because we don't need to sequence anymore, okay? We don't need to amplify anymore. Just one strand is sufficient to sequence. If you're doing paired reads, then you will have basically your P5 adapter or linker. So this is just names that are used interchangeably. And then you have your sequencing primer sequence here. And you'll have the same thing on the other side. This allows you to do a read in both directions. And this is interesting because you'll see this in a second, how the flow cell is made, but you can basically sequence longer fragments by going this way and that way. And if you remember the paired end reading that you do, then it gives you this part here and this part here, and then you can kind of make your contigs from there, right? Although in this case, you don't even need the contigs anymore because you're able to sequence at such a high speed that you don't really care about building your assembly and all that. If you're indexing your DNA that you're trying to sequence, then you will add a tag here. And this is just your barcode, and each DNA molecule has the barcode here that is specific to it, that is unique. And then what this allows you to do is that now you can sequence tons of different fragments in the same flow cell chamber. Instead of here, with these two here, you would need basically just the same fragment used and nothing else. In this case, it allows you to now do multiple fragments at the same time that are of different sequences. Because otherwise, the machine would get signal of different colors all at the same time, and that's not going to work. So you need these tags. Okay. A few considerations. So you still have to, wait, before I say that. So this technique is used for a lot of things in the lab. It's used to sequence genomic DNA, RNA, microRNAs, which are used to knock down RNA. ChIP-seq, which studies basically protein binding to promoters, enhancers, and things like this. So for example, I have a protein of interest called the estrogen-related receptor. I'm trying to study its effect on cancer. And so I will use ChIP-seq to see where this protein binds on DNA. And then we found mutations in this protein, single point mutations, that seem to be found in clinical patients of triple negative breast cancer of the androgen-enriched type. So that means women that have higher levels of androgen and that get this triple negative breast cancer, very rare. So these mutations, you then put them in your protein, you make a protein that is mutated, and you see how the binding of your protein changes on DNA. What does this tell you? You can compare the healthy with the mutated, and then it tells you how the transcription program changes and everything below that. And if you see, for example, that cell proliferation suddenly increases because your mutated protein binds to these regions, then you can develop therapies to counteract that effect. Okay, so this is what I'm doing right now. You can also do this to just pull down exons and remove the introns. There's many things that you can do. Okay, so that's that. Am I going too fast? Kind of. Okay, you can watch the video at 0.75 or something. So when you have your DNA sequence, you have to cut it into smaller pieces. So we still didn't get to the point at this stage where we can sequence long reads. So you have to cut it to about 300 to 600 nucleotides. You will size fragment your DNA using a gel, just like we did before. And this step also allows you to get rid of DNA fragments that don't have the linkers. So you can do this during the gel. Because you can see basically that your fragments, your linkers do not, let's say for example you stain for your linkers or whatever, you see that they're not there, and so you get rid of those bands. You don't use them. If you want to read more on this, there's the Kia Quick kit that you can use in the lab that is used pretty much every day. I use it every day. It's a kit that you can buy that has a series of reactions that you can do, and they provide you with the different buffers. All you have to do is put your cells there, add buffer 1, and go on from there until you reach the point where you have your fragmented DNA. Then from the fragmented DNA, you repair it using the kit still. You add your A at the end of the 3' end, and then using that A, you ligate your adapters, separate on the gel. When you separate on the gel, then you'll have to separate the DNA, well, not separate the DNA, but then you'll enrich it with PCR and use that to sequence it. The step to enrich it with PCR, it's just to increase the signal that you'll see in your machine. Now, what is this? I'll actually get back to the signal. One problem that happened with this method is the fact that you have these P5 and P7 linkers. Nothing prevents the reaction of the linker P5 being bound to both sides, or the P7 be bound to both sides. So we had to find a way to kind of prevent this, because they're used for different things. And so the way you do this is that there was a Y adapter that was created. This Y adapter looks like this, and it's Y like this because these two sequences don't go together, so they push each other away. And then you have this kind of dihybrid that has these two sequences, and then because of this, you have basically the same thing on each side, but now you can use the specific characteristics of the P7 for something and the P5 for something else. I'll show you what those are in a second. Okay. So how does it work? How do you actually do the sequencing? So let's say you have your DNA that has these adapters. You have a cell which is kind of a slide like this, and you have many of your either P5 or P7 oligos on it. And so when you add your DNA, they will complement to it, and you'll have them basically in opposite sense, right? So you will attach them, and they will attach randomly to the flow cell. Then what's going to happen is that you'll start doing your PCR reaction. And by doing your PCR reaction, you will extend your fragments. So these are just steps that are a bit too specific. You don't need really to care about it. But one thing that is important here is that after you have done your PCR reaction, you'll have a double-stranded DNA. That's not good for sequencing. So what you'll do is that you'll have to separate them, and then when you separate them, the other one will bind to another spot, and then that will make kind of these colonies of DNA fragments that are all the same everywhere on your plate. Okay, because a DNA molecule that is copied here will not travel at the other end of the flow cell. It usually doesn't even have time. It will get attached to the next adapter right next to it. How does this happen? So your DNA, so after you denature it, the other side will kind of bend like this, and then will anneal to the other, the other linker on the other side. So this is why you need two different linkers. So you do your reaction, your denature, it will bend over to the side, and by bending over to the side, it will bind to the other one. You'll have multiple ones like this, and then you have to make them straight again. And you keep repeating this cycle for 35 times until you have a small colony of fragments that are next to each other. To separate them, the P5 adapter has this region that you can break, and you can use that by using a chemical called periodate, and it will linearize the molecule. So when you break it from one side, the other side will just go like this. Okay? Now the sequencing. Questions? Yeah.
So that means before we put our DNA on plate,
yeah.
So we actually have to separate the two strands. So there's a step.
Yes, exactly. We have to denature the DNA to put it into a single-stranded size. Okay. Yeah. So now, yeah.
Are linker and adapter the same thing?
Yeah. It's just a word. I'm using it interchangeably, so you get into the idea. They're the same, right? Yeah. So what's cool about Illumina's technology is that it kind of looks like applied biosystems solid, in the sense that you have these special dNTPs, not ddNTPs, so there's no chain termination. Instead, what happens is that you have kind of this termination that happens, but only for a short period of time. So it's not like the chain is stopped and then you see it on the gel, and depending on the size, that means that's the nucleotide that was added. Instead, in this case, what happens is that you'll add your dNTP that is attached, that has a fluorophore attached to it and a termination nucleotide. And so what happens is that when you add it, synthesis will stop for a millisecond. There is a flash of laser that will be sent to the flow cell. It will return a certain light. When the light comes back, there's a reaction that happens in your cell, which will cleave the termination nucleotide and the fluorescent chain, leaving your actual dNTP that is just a normal dNTP. And that allows for more synthesis. And so you'll add again the next dNTP that is hybridized with a fluorophore and a termination nucleotide, image, cut the fluorophore and termination nucleotide, and continue doing that. So this allows you to do sequencing without having to terminate the chains, and decreases the amount of material that you need. Does this make sense? In previous lectures, we talked about chain termination. In the other case, when you terminate the reaction, you terminate it for good. You don't terminate it just periodically and then allow it to run again. Okay, so now you're imagining this flow cell like this, and it has all these DNA molecules that are clones of each other that are in kind of groups. So then how do you image this? What you're going to do is just basically throw laser at everything. And when you throw laser at everything, the fluorophores will emit light. And when they do, you'll see these dots on the plate. And then each dot will give you the sequence of that specific area. Okay? And obviously, it's integrating signal from multiple things. So it has to do some calculation based on the size of the circle, for example. And if there's an adjacent circle to it, if it's a different color, then it has to make sure that, that was not a mistake and things like this. But it's still not perfect. Actually, they're improving this. One way to kind of prevent these problems is by having these adapters here that are barcoded. This allows you to have different, you can index your DNA sequences, and you can sequence different fragments at the same time. So that's that. If you're interested in chemistry, this is your DNTP, and they basically just added the fluorophore and the nucleotide, the termination signal. And so what you can do is just break this here, and you can use simple chemicals to just break this bond here. And that will basically turn this whole complex into an NTP, DNTP. Yeah?
So does the bound nucleotides only fluoresce when they bind, or do they wash all the unbound ones out before they image? Like, how do they make sure that the unbound ones don't interrupt the signal?
Do they flush it? So basically tell me if they remove the fluorophores every time.
Yeah, like the ones that didn't get bound. Or is it just that they have to bind before they fluoresce?
Yeah, they have to bind before they fluoresce.
Okay.
Yeah. They have to bind before they fluoresce. I don't remember exactly the mechanism through which this happens, but I can definitely tell you about this next lecture. I can dig deeper in this. Okay, and then your plate kind of looks like this. And you have different colors everywhere, and then you can just figure out the sequence from there. Okay. What this technique does is that it gives you it allows you to do parallel reactions. So you can have tons of fragments that are read at the same time. And it's highly not reproducible, but if you're getting signal from different fragments that are in a colony, that signal might be, for example, there might be a mistake in one of the strands and not the others, and so the signal is integrated together and gives you a final result with the highest signal being the nucleotide being incorporated. Because you always have mistakes because nucleotides are specific, but sometimes the wrong nucleotide might be added. Okay? So the linkers. The linkers, I think I just explained this slide here. I was not supposed to get into this, but I basically explained it earlier. So this is just the 3 generations of linkers or adapters. And the first one is just to do a one-direction read. The other one is to do 2 reads, one in each direction. And this one here is to specifically barcode each DNA molecule so that the sequencing can then be specific to that barcode. Well, ion torrent. This, I think Alex talked about this at the beginning of the semester. So the ion torrent was the pH-sensitive method. So this is again a second generation sequencing method. It's probably the cheapest system, but still the least used. And basically, it's a small machine that costs about $50,000. So it's not that bad compared to the other ones, which are in the million range. Illumina, I think, is like 900K or something like this. So this one is quite cheap. So this resembles the pyrosequencing. The way this works is that you have a DNA polymerase in a semiconductor, and this semiconductor will detect the increase into the hydrogen in the protons. And what it will do is that it will basically act as a pH meter, to simplify it. So as the protons are released from the chain synthesis, they will create a signal, and that signal is integrated as pH. Now, how do you separate the different nucleotides? Well, you will be adding them at different times. So you add an A if there is an increase or decrease in whatever, an increase in pH. then you will decrease in pH. Then you will detect it as that nucleotide being added. If there's 5 of them that are added at the same time, 5 A's in a row, then we get back into that initial problem of having a limit of detection. You know, when your pH gets super low, there's not much more you can go, right? You can go a negative pH or something. So there are limits for this, so which are basically the same. This figure here just shows you, again, the process, but it's quite repetitive. Why is it repetitive? Because these technologies focus on the same idea. You have DNA, which is your genomic library. You break it into pieces. Then you will prepare these beads that will hold your DNA based on the adapters that you add on each side. And then you will do a PCR reaction to amplify your signal, right, to have more copies of the same fragment. And then you will do your sequencing, in this case, through this technology. Okay? And again, where do you do this? You do this into these small wells, which are semiconductor wells. Clear? The only thing that changes in these technologies really is the last step. The rest is pretty much the same. What else? So one thing that's interesting about this is that you're working with silicon chips. You're not working in some sort of buffer or whatever. And so, well, not true, but you're working with these chips that can directly detect the pH. So obviously this makes the machine much more smaller, much more portable. Obviously microwave is not portable, but it's not as bad as the Illumina machine, which is kind of, you know, super big. So you can move this and use this on the field directly. This is not really relevant for people that work, for example, in labs, but people that go to different places. Instead of storing your samples until you get back to the lab and affecting them by changing temperature and all that, you can directly get to the work there. So that's kind of one advantage. It just has the same limitation. You can't distinguish when you get to a high amount of protons released. You start saturating the signal, and you cannot see if you have more nucleotides of that type added or not. At some point, there's no difference. This one is quite cool. So these are just the evolutions of the chip that's being used. But basically, when they started, they had about an output of 40 megabytes of data. The runtime was about 1 to 2 hours, and you could read 400 base pair long fragments. And that gave you up to 0.6, so 600,000 base pairs, bases that are read from one single chip. Go forward to July 2013. Now you have a much higher amount of sensors on that chip. So this is kind of crazy, this amount on a chip. You produce about 100 gigabytes of data. This is a lot in 2.5 hours. The size of the reads are smaller, but it allows you to do a lot more reads. And so you can read about 330 million bases in one cell. I don't know how they fit all that in there, but this stuff is kind of all patented. Some stuff is online, but obviously other stuff, they omit it. If you want to look at this stuff, actually I can show you right now. I don't know if people are interested in patents. So you can go here, you can do PacBio, for example, and just read, okay? See who owns the patent, when it's gonna die, and if you wanna steal it or something, go for it, okay? So this is one thing. Interestingly, unrelated to the course, but, well, actually it is. If you're in genomics, you might be inventing stuff like this in the future. Patents, you can pretty much have all the information in there to reproduce what they're doing. So you can kind of play with the patent, modify it a little bit according to what's legal, and then build your own stuff, right? So this is something interesting, and you can read the patent for pretty much anything that exists in the world that is kind of patented right now. If you're working in a research environment at a university and you make discoveries, the university takes part of that patent, and I think they take like 50% or something like this, okay? So this is a consideration that you have to take if you make discoveries or if you do some work in the academic area, okay? Usually the university takes a percentage, the director takes the other percentage, and then you end up with something super small, okay? So you have to think about this when you're doing work, especially if it's work that's creating stuff and that can lead to patents. Totally unrelated, but I'm telling you just in case you make the mistake. Because I've made it before, but don't want to get into this right now. So that's all right. So third generation sequencing. This is cool. This is much cooler than the other two because now you just need one single DNA molecule. You don't need to do amplification. So if you have one fragment, you can use that fragment to sequence. This means that you need much less sample. So if you're doing the Neanderthal's genome, you don't necessarily have a lot of it. You do have the whole bone, but how much of it is actually good DNA that you can use for sequencing? So you're going to try to use third generation sequencing, which gives you the opportunity to work with a single strand DNA molecule that is not amplified. Obviously, every time they keep decreasing this cost per base. I don't know how far they're going to go, but limit is 0 there. So at some point they'll stop. I think now it's like a cent per, or like 0.01 cent per base or something like this, okay? You can sequence in minutes instead of days. So that's one thing. The sample preparation is super simple. You always remove stuff that you can optimize. If you remove the PCR reaction and the flushing and all that, then you end up having less sample preparation to do. Although this is kind of true and not true, because if you have a single strand of DNA, you want to be super careful with that. So that kind of puts these measures in place that you need to take care of your sample. And it allows for long reads in a shorter time. Usually when you optimize something, no matter what it is, decrease the time, increase the speed, and decrease the cost, okay? No matter what you're increasing, this is what you're changing, so this is the case for this. Two techniques that we're going to talk about super quickly. To not get too much into the details, but you have PacBio, Pacific Biosciences, which was at one point, I think it was going to be purchased by Illumina. And then there was an issue in the US about this due to the monopoly of the sequencing, let's say, field. And so I think they even went to Congress or something like this. So, people don't want one company to own the whole market, and Illumina is still killing it. So they still have the monopoly. So I don't know what that was good for. But they tried to acquire, I think, Pacific Bioscience for like $2 billion or something. Now they're worth like 50. Okay. So Pacific Bio, aka PacBio, uses something called a zero-mode waveguide, which is just this special cell here inside a plate. Now this plate here is 100 nanometer high, so this is kind of super small. You can't even see it with your eyes. And you have to fit in one DNA molecule in it and one polymerase. So this is one of the complicated things. And the detection limit is like 50 nanometers, maybe a little bit less. So this is some super advanced technology. Obviously, not everything of it is on the patent. Some of it is hidden because you don't want other people to reproduce your stuff. Even though you patented it, people will still do it. So we don't know 100% what is happening in here, but this is what they say is happening, at least. And they use the single molecule real-time sequencing. So you're basically sequencing using a single molecule that looks exactly the same as the one that we talked here, where you have your fluorophore attached to the DNTP. The only difference is that in here you don't have the stopper. So termination does not happen, just keeps going, okay? So this is quite precise, although they have some problems because sometimes the speed at which this happens kind of messes up the signal. So it's not the most precise technology, right? And so you have these nucleotides that are labeled on the terminal phosphate. This way it prevents the blocking the chain here. And then they are trapped in this, what they call a waveguide, for enough time to be detected as the nucleotide is being incorporated. So this is just based on the size of these, the shape and the size of these kind of wells, these 100 nanometer high wells. If you obviously change the size of this, then this does not work anymore because the DNA will be kind of moving, and that will affect the signal recognition. So some points about this. First, the nanostructure provides a confinement space for the reaction. This is what I just told you. The only problem is that it's super small. So what happens is that, so before I get into that, it's in the zeptoliter size. So that's quite small in terms of volume, 10 to the minus 21 in each one of those small spots. So you can see how hard it is to fit the polymerase and to fit your DNA molecule. This is one of their lagging problems that they cannot improve on the technology. You have these phospholink nucleotides, so that prevents steric hindrance. They made these nucleotides here in a specific way so that you don't prevent anything from happening, because it's super tight in here, right? So you want to have everything placed in a way that will allow for the detection. You see this line here? If this nucleotide was, for example, a little bit higher here, then it would not be detected, okay? So this is how precise it has to be. I don't know how they did this, but this is some crazy work, okay? You don't need to wash between the cycles. As the nucleotides are added, they're detected. And it allows for real-time sequencing of molecules up to 100 kb reads. So 100 kb reads is not in one well, okay? It's in the full plate, right? They were able, so they started their work when they built the startup using E. coli genome, and they were able to get 586 bases. Now they're able to get even more than that, and they can read up to 100 kb. So this is a huge increase, okay? Now, getting the human genome, the title of this lecture. The filling the gaps, the paper that came in 2022 to fill in the gaps for the human genome used this technology, and it's called the T2T project. Okay, this is what filled the genome, the remaining portions of the genome, so they used Pacific Bioscience. What else? Yeah, so this I just said. One of their limiting step in the improvement of the technology and why they're not killing the market, it's because it's very hard to put the DNA polymerase and the DNA into the waveguide. So this is one limiting thing. What they're trying to work on, the improvements, is to actually direct the DNA into the chamber using different physical properties of the DNA molecule. But again, I'm not so, it's hard to find information on this. They can just find it on the news that they say, but you can't get into the details of what they're doing, right? Because this is all protected information. They made some improvements to it. Now you're able to read 20 base pairs in one go. On average, some of them are even up to 30,000 base pairs. So if you have a cell that has these zero-wave guides, that has 15,000 zero-wave guides per cell, then you multiply these numbers, you can read up to 10 to 20 billion bases. So this is a lot more than the human genome. Okay, so one cell can take care of the human genome pretty well. Yeah?
So are they using this to fill gaps because it reads so long? Is that why they picked it?
Exact. And also, I have to remember this. Let me just look at this so I can... the problem with... So the gaps, when they were created, it's because of the issue of the contigs, right? And so you were jumping from one place to the other. And so now if you can have longer reads, and the reads are happening as they are incorporated, then you don't have that issue of figuring out if this repeat is from this fragment or that fragment. Instead, because you can read something super long, and you do it in real time, then you don't have that issue for the gaps. But very good question. What else? Okay. I think we're almost done. There's 4 slides left. Oxford Nanopore, this was brought up, I think, second lecture. This is another technology that is of the 3rd generation type. This one here works by having a lipid bilayer. And the lipid bilayer, it has a pore in it. and this pore will allow DNA to go inside some sort of cell, and it has a rotor or motor protein here that will kind of pace the DNA while it goes inside the cell. And as the DNA molecule goes inside the cell, what happens is that there will be a change in the electrical properties of the solution in here, and that will be detected as electrical signal, and will give you these, I guess, pico or nano amps, and that will tell you it's specific for each nucleotide. Okay, this is the only technology where instead of building, you're kind of separating. So the others, you are adding nucleotides or you're ligating a probe. In this case here, you're breaking the DNA into two, and only one strand goes in, and nothing happens to that strand other than as it passes the pore, there is a change in electrical field, and that's what sequences. So this one is kind of special. Again, this here allows for a small sequencer, so you can, these pores are found on a small chamber, right? So these microfluidic chambers, which are small like this, and you can basically use them, you can kind of detect these signal changes on the field because the machines required to read that are super small, okay? So you can take this with you anywhere you want. So the initial strategy was not this though. The initial strategy was that they were allowing the DNA to go inside the cell, and then they were cleaving the DNA as it goes through the pore, and every time there's a nucleotide that was removed, those nucleotides were creating a signal, and that signal was being kind of assessed. The problem with this is that some nucleotides would run away and they would not be detected, and so that was changed, and I'll tell you what they did to change it. But let's start talking about the pore first. So the pore, they're all biological. So this gives you an idea of how people are thinking. So they took a pore that already exists in bacteria called hemolysin, and then now they're using this R9 pore, which is based on this gene here, highly mutagenized because they had to change some properties of it. But basically, you can think of this as, it seems very complex, but in reality, you can think of it and say, okay, I have a DNA of double-strand DNA. Let's just open up the DNA, pass it through a pore that would allow one nucleotide to go in at a time, find a pore that allows you to do that, and then create this chamber with this pore. And now you just need a signal, right? So you need some sort of solution that is able to detect current, and you plug it. Very quick. But if you remember this question here, right? I'm talking about double-stranded RNA molecule where you have 10 chromosomes, 12 billion base pairs, but you're in 1990, so you can't really use this technology, right? But if I told you you're in 2024, then you can use this to describe it, and you would go as follows. So you would say, okay, I have my 10 chromosomes, I will separate them, each chromosome by itself, and then I will use this technology because it's able to read 10 to 20 billion bases. I will pass it through the pore, and that pore will give me an electrical signal that is specific for the nucleotide, and then things like this, okay? And then I will assemble all that data, and I have the sequence. I'm going super fast, but you can expand on this, okay? So this is one example. Last two slides. Okay, here I'm gonna get a little bit into the speculation because these companies obviously have to market for themselves. So they do claim some stuff. The first thing, before I get into the claim, so when the single-stranded DNA molecule or RNA molecule or whatever molecule is passed through, the read seems to actually happen with two nucleotides and not one, okay? So they say it's one nucleotide, but it seems to be two nucleotides, so that's the first thing. And so the signal is not as simple. It's like the solid system, kind of two nucleotide system, so you read two out at a time, and you need some sort of complex analysis to differentiate these here, okay? Because it's a two, every time you add a nucleotide in the system, you're adding more complexity. So that's one thing. They claim that they have a 90% accuracy. I say claim because it's not always true. We've used these technologies and it sometimes does not get to this point. But if you read on their technology when they try to sell it, they claim this is it. But it's kind of, when you're, for example, building something, you know how you built it. So you know all the problems with what you built. So when you're testing it, you can't be the one testing it, because if you test that thing, you'll be biased and you'll just take the route that you know will work. But if you take that technology and make other people test it, for example, me who's trying to sequence, or other labs, will not get to this 90% accuracy. But you, because you know how your thing works, and you've been using the same samples nonstop, you've optimized it for those samples. Obviously, I'm making it super simple here, but in reality, they did more tests. It's just this 90% accuracy. It's like this. Yeah, Alex.
Is that 90% per read or 90% for like a given sequence?
90% for a given sequence.
That's kind of trash, isn't it?
It's not. It's actually. Well, the problem with these things is that, so if we're talking, for example, about Illumina, and you do sequencing, the first 100 base pairs, the bases are useless. And so if you do a read of 700, the first 100 come out as being like kind of mixed up signals, right? And so that lowers the accuracy to much lower than 90%. But for this 90% accuracy of the whole sequence, I mean, compared to Illumina, it's pretty good. I'm not sure of the specifics why it's 90% accurate. Actually, I can take a look into the reason why the first 100 reads are not. So for Illumina, I know the polymerase at first is very shaky, and so it's not the sequence that gets polymerized is not as perfect. But for Oxford Nanopore, I have to check why this is not good. But if you have a 90% accuracy for each nucleotide, that gives you a 90% accuracy for the whole fragment, right?
Yeah, but if it's like, if no matter how many times I read the same DNA sequence, it's going to be the same errors, the same spaces, then I can't be confident.
Yeah, I will read on this, and I will let you know the specifics of this next class. Okay. Questions? I need water. Okay, a few more points. They're able to read about 300 bases per second. They're hoping for 500 per second, and obviously their vision is 1,000 per second. If they get to this amount, they will be able to read, I think, 1 billion per 3 seconds or something like this. This is crazy, okay? This is like crazy amounts, but obviously you have to sell the vision for the investors, and then you have this part here, okay? What's the role of the motor? The motor is not to put the DNA inside. It's to slow it down because it goes way too fast in, and so you want to slow it down so you have enough time to measure the electrical signal. And the motor used this E8, which is a modified helicase, probably a modified helicase, but you don't know what it is. It's called E8. When you're doing research and you're working on a compound that you don't want to tell people about, when you publish, you name it C75, right? So you know what it is. People don't know what it is. E8, I don't know what it is, okay? But probably a helicase which unwinds the DNA at a specific speed. At last, this is the graph that comes out of it, and then the computer makes sense of this picoamps and tells you which nucleotide was added. That's it. Any questions? No? Good. Anybody coming to the office? Sarah? Yeah. Okay, cool. So I'll see you next week. I'll extend the assignment for number 3 for Monday. I'll try to figure out the grades, why they don't appear online. I mean, I just tried, it didn't work. Maybe I'll just send it to you by email. Okay, it's going to be easier. And then you can update it and then go on from there. And when you get your grade for the 3rd one, you'll know if your incorporations now make sense, and you can use that same style for everything that you're doing. You should be good. Okay? Take care, guys.